So we have discussed about most of all the organelles in the eukaryotic, like endoplasmic reticulum, mitochondria, plastids, what else, ribosome, vacuoles, lysosome. Now we are talking about the last and the main important one, which is nothing but the nucleus. Now let's get into the video. Nucleus is first discovered by Robert Brown in 1831. Robert Hooke found the dead cell. Okay, he is the one who find the dead cell first. That is Robert Hooke, but nucleus is found by Robert Brown. Now we will discuss the nucleus with the structure. Basically, this nucleus is made up of inner and outer layer, and it is the brain of the cell. Okay, it controls, has the activity, and it has the DNA. So it is called as the brain of the cell, and it is made up of two layers. One is inner layer and another is outer layer. Within this inner and outer layer, there will be a space, right? It is called as the perinuclear space, and the size is of 10 to 15 nanometer. So we have two layers of the nucleus, which is inner and outer. The space is called the perinuclear space. Then we have nucleopores, which are there between these layers. Whenever the fusion of these layers is happening, say here the fusion has happened, so we have some space. This space is called as the nuclear pores. Okay, these pores are called as the nuclear pores, and these are helping transporting the RNA and protein in either side, that is from nucleus to the outside, from outside to the nucleus. Okay, that is the function of this nuclear pore. And we have nucleoplasm. So this is that a surrounded area in the nucleus is called as a nucleoplasm or nucleomatrix. Within this nucleomatrix, we have nucleolus, which is the site of ribosomal RNA synthesis. Okay, now ribosomal RNA synthesis is done in the nucleolus, and in each cell the nucleolus is differ. Okay, if it needs the high protein synthesis, the nucleolus will be in high in number. Okay, now we have something called as chromatin in the interphase nucleus. What is the interphase nucleus? The nucleus which are not dividing, it is called as an interphase nucleus, and in that we have the chromatin structures. These chromatins are first found by Fleming. Okay, Robert Hooke found the nucleus, but chromatin was found by Fleming. He found it when the nucleus is stained by the basic dye. As we discussed about the gram positive and gram negative bacteria, here also some basic dyes are used to stain it, and from that we found out this chromatin. These chromatins are nothing but the network of nucleoprotein fibers, and it has the DNA histones. Histones are the basic proteins, we could say. This and all we will be talking in detail in the 12th chapters. Histones and non-histones and RNA, these are the materials found in chromatin. So this is a general view which I have simplified. What are the things given in NCERT? You have to remember these things, that will be enough. That is inner and outer membrane, perinuclear space, nucleopores, nucleoplasm, nucleolus, chromatin. And the main part is this endoplasmic reticulum will be attached to the nucleus, and also it bears the ribosome. So as you all know, when it bears ribosome, it is the rough endoplasmic reticulum, because we have the ribosomal RNA synthesis happening here. That's why this endoplasmic reticulum is closely attached with the outer layer of the nucleus. Now we will talk about some few information given about DNA. So the DNA in the single human cell will be of 2 meter long, and it will be like spread out with the 23 pairs of chromosomes. As you all know, we have 23 pairs of chromosomes, so 46 in total. So the DNA will be distributed along all this, and it is of 2 meter. And we have some different types of chromosome based on the position of the centromere. We have metacentric, submetacentric, acrocentric, and telocentric. All these things we will be discussing later in the upcoming molecular chapters. Okay, molecular inheritance, those kind of chapters. So I have given the basic idea, and we have given about microbodies. These microbodies are nothing but some minute vesicles. Okay, and it is found in both plants and animals, and it is a membrane bound organelle. So with this we are completing this chapter. So we will see the questions for this page. Nucleus is found out, discovered by whom? chromatin discovered by whom? What are the function of nucleopores? Size of perinuclear space. Place of RNA synthesis in nucleus. What is the size of a DNA in single human cell? Does microbodies have membrane or not? So with this, we are completing this chapter successfully. I hope all this video will be helpful for you because I have included the questions in between. I haven't given it in the forms of MCQ because I hope you will be able to answer these questions even without options. So that's why I will make an MCQ video or like Q&A video for this chapter covering all the topics in a single video also. Keep revising. We'll see you in the next video. Please try to watch these strict videos. That will be really helpful for you. Thank you.